Yasmin, go do something.
Maddie face.
Yasmin, go take smiley face over there.
No, Maddie face. Mad.
Yes,
Probably want some chicken wings before you do your lecture?
No, it's live. We're live. I ain't got no time for wings. It's recording.
Oh, we're hungry.
Well, then eat.
Me too.
Here we go.
Me too.
I mean, you're funny, Mommy.
All right, guys, give me 1 second. I'm gonna. Well, hold on. All right, first things first.
They know us, so.
Give me an audio and a visual. How is the live stream feed? How's the video? And then somebody give me a sound check. How's the audio?
I want some.
1 second. I'm just gonna post something to the classroom, and then we'll be good.
It's too hot. It's too hot. It's too hot.
So what do you want to do? You want to take a piece off?
Color. I want color. Color, 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 color. Happy face.
Hold on, this Bubba's working, Mama.
Yasmin,
Yes.
I need you to be quieter.
Say hi to Bubba's friend.
It's not funny.
Look.
Go say hi to Bubba's friend.
Look. Mommy, look.
Happy face.
Happy face. Give me more happy face. Okay, good.
Okay, the announcement on the Google Classroom is posted, so whoever wants to join us can. All right, let me see here. Jack, what's up, buddy? Audio's good? Video's good? All right.
Hey, Baba. I'm stuck. I'm stuck.
All right, whoever's in the live chat, make sure I know where you're at here. Say a hello or something. You guys just want to be all inconspicuous. Thanks, Mama.
Say hello, you say hello to the camera.
Just mean you want to say hi? Can you see? Look. Pop your head up. Pop your head up. You
Hi,
hiding now?
friends.
Baba. Huh?
Really, Yasmin?
I just saw that.
Go over there. Ridiculous. Alright, let's see here. Jack's already got questions. He's all impatient. Alright, so my original plan was to actually have some structure with this live stream. I was going to go through everything in the slide presentation in chapter 2, and then after each section just ask if anybody has any questions on that section. So what I'll do is, if you have any questions, just hold on to those. I'll come back to them. But I'll just go through all of chapter 2, section 1, going through the slideshow, letting you guys know what's important and what you need to know. And then if you have questions on it that are relevant to section 1, then ask. All right, so I'm going to start with section 1, which was Intro to Chemistry. Now, a lot of what is in section 1 should be reviewed. You guys should have learned this in physical science. So hopefully this is your second or third exposure to it. So for section 1, it's called The Nature of Matter. Make sure you're good with the atomic theory. You understand that the atom is the basic unit of matter. Make sure you understand and can identify all of the subatomic particles and their charges. So protons are positively charged. They're in the nucleus. Yes, Yasmin. Neutrons are also in the nucleus, but they have no charge. They're neutral. And then electrons orbit around the nucleus. They're negatively charged. And those are the energy molecules of the atom. So that's kind of like the basics part of that. All right, the specific thing that you have to know as it pertains to subatomic particles is valence electrons. Make sure you know what they are. They're the outermost electrons in the atom. They're actually what gives atoms their particular identities and characteristics and properties. Yasmin, come on. All right, so I'm just going through the slide presentation. All right, for isotopes, make sure you know what an isotope is and be able to provide an example. So an example would be like carbon, how it has 3 versions of carbon. And the only thing that differs in isotopes is the neutron number. So the proton number, the electron number, those are the same. The neutron number changes. So carbon has carbon 12, 13, and 14. Carbon 12 has 6 neutrons, carbon 13 has 7, and carbon 14 has 8 neutrons. So that's a good example of an isotope. And of course, different isotopes have different properties. And make sure you're just familiar with the periodic table. Make sure you know the noble gases. Make sure you can identify things by their atomic number, their chemical symbol, their groupings, stuff like that. As it pertains to the most common elements in living things, you have carbon, hydrogen, oxygen. Those are the top 3. Then you have nitrogen and phosphorus. And all organic compounds contain at least carbon, usually carbon, hydrogen, and oxygen. Definition of a compound: any substance that contains 2 or more elements. So water, for example, that's H2O, that has 2 hydrogen atoms and 1 oxygen atom. Therefore, because it has 2 different elements in it, it makes it a compound. Carbon dioxide, CO2, also a compound. Salt, table salt, NaCl, sodium chloride, that's also a compound. I gave you guys an example last week of O3, which is 3 molecules of oxygen. That actually is not a compound. Yasmin, that's enough. Come on. Be nice. I'm busy. Don't be rude. Make sure you are able to contrast ionic and covalent bonding. So sodium chloride is a good example of ionic bonding, where the sodium and the chloride interact in a funny way, and they actually transfer electrons to one another. So in this case, sodium donates an electron to chloride, making the chloride more negative and making the sodium more positive. Covalent bonds are bonds shared between atoms where electrons are shared between them. There are single, double, and triple covalent bonds. We talked specifically... Yasmin, that's enough.
Yasmin, let's go to your room.
I gave you guys the example of water as having single covalent bonds between the hydrogen, each of the hydrogens, and the oxygen atom. And that was a good example of covalent bonding. Let's see what else here. All right, so that does it for section 1, actually. So let me go ahead and see what's going on in the live chat and see if there's anything there from section 1. If you have a question on section 1, anything I talked about, go ahead and ask it now. Jack, I'll get to the buffers in section 2 when I talk about solutions. Regan, you do not have to know van der Waals forces. I'll let you guys worry about that when you take chemistry next year. Abby asks, will there be any questions on adding up protons and neutrons to a specific atomic mass? Nope. We won't be doing anything like that. Because then that would give you your atomic mass, and it's not necessary. Jack, we'll talk about hydrogen specifically as it pertains to water in the next section. That's where your question becomes more relevant. Liv asks, will we have to memorize stuff from the periodic table? I mean, you wouldn't have to memorize it because you're allowed to use your periodic table on the test, but you can't have anything written on your periodic table. So it has to be just a plain periodic table, no writing, no notes, nothing like that. The only thing you really need your periodic table for is if you needed to refer to it to see what the atomic number is or something like that. Yeah, Liv, swag indeed. Yes, swag. Any other questions from section 1? All right, if you guys do have questions on section 1 that pop up later, just go ahead and ask them at the end when we do our Q&A. All right, Jack, so it's not really about being different between compound and Covalent and ionic. Okay. So sometimes when elements interact with each other, different atoms of different elements, they interact with each other in a way where they start to reform different types of bondings. If the bonding type that they're forming is covalent, that means they're sharing valence electrons. That means, for example, take water, for example, right? Water has one oxygen and then it has two hydrogens in it. Well, there's a chemical bond between the oxygen and then the two hydrogens that are bonded to it. And the bonds between each of the hydrogens and the oxygen, those are covalent because they share valence electrons. The valence electrons are not being transferred from one atom to another. They're shared. They're kind of around both of them. So when the valence electrons are shared and they spend time around both different atoms, they're called covalent bonds. Now, if the valence electrons are being transferred from one atom to another, so the electrons from this atom are going to this atom, we call that an ionic bond. So the valence electrons are being sent over, they're being transferred to the other one. Now that makes the one that's receiving the electrons more negative, and that makes the one that is losing the electrons more positive. So there's a good visual actually in the book where it talks about the different charges that these ions are formed when they gain or lose electrons. And I think that's really where you want to be. You want to be able to see what the valence electrons are doing. If they're shared, they're covalent bonds, and if they're being transferred, so they're being like donated and accepted, then those are ionic bonds that are formed. Hopefully that answers your question. If you want more clarification, you can ask at the end of the live stream. All right, I'm going to move on to section 2. This one is on properties of water and other solutions. All right, so water. At a chemical level, water is H2O, 2 hydrogen atoms and 1 oxygen atom. Hydrogen has the atomic number 1, so that's 1 proton, 1 neutron, 1 electron. There are 2 hydrogens in water, and oxygen has the atomic number 8. So 8 protons, 8 electrons, 8 neutrons. Now, if you think about the way that water works, water has some really, really unique properties, and it comes from the types of bonds that interact within the water molecule itself. So the first thing to remember, water is a polar molecule. So what exactly does polar mean in this case? Polar means that one side of the water molecule has a negative charge, which would be the oxygen side. The opposite side of the water molecule has a positive charge. So one side is negatively charged, the other side is slightly positively charged. So they're differing in that respect. So we say that they are, they form these covalent bonds, right, between oxygen and hydrogen, but they also have these polar properties, which is where water gets all of its uniqueness and what makes it different. So not only does water have covalent bonds between the hydrogens and the oxygens of the water molecule, there's also hydrogen bonds that form between separate water molecules. So if this is water molecule A and this is water molecule B, they naturally have an affinity for each other. This is how water gets that cohesive property. So it's the oxygen on this one, which is negatively charged, and it's the hydrogens on this one, which are positively charged. They attract to each other. And that's how we get things like surface tension, adhesion, cohesion, capillary action, high heat capacity, everything we talked about in our discrepant events from the lab last week. So I think it's worth it for you guys to take a look in the textbook at the chemical composition and the bonding things that are occurring with water. It's in the slide presentation. It's also in the textbook. So slide number 12 in the slide presentation, if I can just pull it up here, talks about cohesive properties in which water beads together, water molecules stick next to each other. You guys remember in the discrepant event when you're putting water drops on the head of a penny, you see that the edges of the water bubble kind of spill over the edge, but they don't necessarily fall over right away. They kind of cling on to each other for a while. That's the cohesive property of water, by which those hydrogen bonds are actually holding water molecules to one another, so giving them that cohesive property. It's also responsible for the surface tension that is created on the top of the water level where the meniscus is. So how was the basilisk lizard, the Jesus lizard, able to kind of walk or run on top of water? Is because water has this cohesive surface tension property and also the meniscus, how it does that little bowing effect, right? On the edges, the water kind of comes up and then it bows downward at the bottom because that's gravity pulling down water, but the water on the edge is kind of pulling up on the surfaces of the container. So water has some really unique and interesting properties, all of it stemming back to the cohesive, the adhesive properties, the capillary action. All of it goes back to the fact that water is polar and has these hydrogen bonds that form as a result of that polarity. Slightly positive on one side, slightly negative on the other. The next slide talks about adhesive properties, capillary action, and then high heat capacity, how water can actually absorb quite a bit of heat energy and then stay warm for a long time, which kind of describes why coastal areas usually have very, very pleasant weather, even during the colder months. Next is the section on solutions. So you all should know the difference between a mixture and an actual solution. Oftentimes mixtures are, you have some form of a compound inside of another. They don't actually have a chemical interaction. That involves a solution that has like a solute that gets dissolved into a solvent. So that's really what a solution is, when you dissolve a solute into a solvent. So taking sugar, dissolving it into water, you now have a sugar solution, where the sugar is completely dissolved and chemically interacted with the water, which is very different than a mixture or a solution, or a suspension, I should say. So if you guys remember, we talked about oil and how oil is non-polar and water is polar. Because of that, they actually repel each other. So you'll always see oil sitting on top of water, and that is because of the differences in their polarities, and also the fact that oil is also less dense than water, so it floats to the top, and then water sits at the bottom. So that's I think on slide number 15 of the slideshow. And then it goes on to talk about hydrophobic and hydrophilic properties. We describe hydrophobic as being water-fearing or water-repelling. Hydrophilic means it attracts or likes water. So fats, oils, waxes, unsaturated, saturated, all those types of lipids that we talked about, those are all hydrophobic because they are non-polar compounds. Now, if something else is polar, then obviously it has an affinity for water because water is also polar. Slide number 17 is the section on acids and bases. Make sure you're familiar with the terms. So what makes something acidic? Anything with a very low pH, below 7. Anything with a pH above 7 is a base or is alkaline. A neutral substance means that it has a pH of about 7. Now it's important to note that when you think of acids, think of H plus ions. In fact, those H plus ions are indicative of an acidic solution. And the more H plus ions it has, the more acidic it is. So say there's a substance that has a pH of 6 and something that has a pH of 1. They're both acids, but anything with a pH very, very low, like 1, has a lot more H plus ions than OH minus. So anything above 7 that is a base has a pH, maybe say 10 or 12 or 13. That means there's a lot more OH minus ions, hydroxide, than the H plus ions. Now, what does this do for neutral solutions with respect to those ions? They're equal. So take, for example, pure water. Pure water has a pH of 7. That means that pure water has the same number of H plus ions as OH minus ions. That shows that they're equal, so that's why they are neutral. On slide 18, it talks more about, different examples of items or typical compounds that you come into contact in your everyday life. So cleaning products like ammonia cleaners, oven cleaners, detergents, soaps, they're usually alkaline or basic. They range anywhere from a pH of 10 to 13. Milk of magnesia has a pH of 10. Baking soda has a pH of 9. Marine water, like seawater, has a pH of about 8 to 8.5. Pure water, distilled water, has a pH of 7. Most body fluid, so we're talking about like really common things like blood and urine. It's not gross, we're talking about life characteristics and properties here. So they're usually pretty close to neutral too because they're mostly water. So that would make sense that they're somewhere between like 6.5 to about a 7 pH. Black coffee has a pH of 5, so it's slightly acidic. Tomato juice, orange juice are a little bit more acidic, pHs of 3 and 4. Lemon juice is a pretty acidic compound, pH of about 2. And then gastric fluid, which is your stomach acid, which is obviously very corrosive. It helps digest food and break it down, has a pH of about 1. And anything at like 1 or lower is considered a superacid, and they are very, very corrosive and dangerous. Hydrochloric acid, HCl, is the chemical formula for your stomach acid. And then, Jack, you were asking about buffers. You should know what a buffer is. Basically, what a buffer does, a buffer serves to kind of balance acids and bases. So let's say, Jack, that you were testing a solution and you saw that it was very, very acidic, like a pH of 2, and you wanted to neutralize it. What you would use is you would use a very strong base. So if something has a very, very low pH, like 2, and then you have a buffer that you want to use to bring this up to about a 7, you would have to use a strong base to kind of level it and neutralize it to about the same point where they'd be at about 7. So we call those buffers. A lot of people, when, say for example, something is very acidic, maybe a pH of 3 or 4, they'll add baking soda to try to raise the pH a little bit to make it more neutral. Because of course, lemon juice, tomato juice has a pH of 2 or 3. If you were to add a basic buffer, that would level it out and make it closer to neutral. So that's what buffers do. And you can also use a buffer to take a very high pH and bring it down. You would do so using a very strong acid. So it would level out and eventually end up near about a pH of 7. So that's what buffers are for, to either neutralize solutions or to kind of counteract strong acids with, strong bases, or vice versa. I don't think there's anything specific more than that on the test when it comes to buffers. In fact, some of the forms may not have a question on buffers. All right, so that wraps up section 2. If you guys have any questions, go ahead and ask in the live stream chat. I'm going to go ahead and I'm going to look to see if anybody has any other questions. Regan asked, do we have to memorize any of the liquids and their pH? Well, it's good to know some examples of common substances that you come in contact with regularly and their pHs. So try to remember it this way. Any type of fruit juice, anything derived from like tomato or lemons or oranges, those are acidic. So their pHs are usually below 5 or 4. Like lemons, for example, have a pH of 2. Tomatoes a pH of 3. Oranges pH of 3. So they're pretty acidic. For neutral solutions, just make sure you know like pure water, blood, urine. Coffee has a pH of about 6. I mean, these are things that you typically come in contact with on a regular basis. Baking soda is a base, has a pH of 9. Ammonia, pH above 10, about 11. Oven cleaners, detergents, household soaps, stuff like that, they're usually pretty basic, pHs above 10, 11, 12, and 13. Jack, so the difference between a mixture and a suspension, you can actually see what a suspension looks like. If you see particles in, say for example, water, you see little floaties, you see little particles, that means those molecules are not dissolving into the solution. So that would be a suspension. So, take a bunch of little pieces of gravel, dump them into a beaker of water. That will never dissolve into the water, right? That's always going to be a suspension. Or take, for example, a mixture of oil and water. You can mix the oil and the water artificially, like mechanically you're doing it, but eventually they separate and they form a suspension where the oil's on top and the water's on the bottom. They never commingle, they never interact, they separate, and that has to do with, one being polar, the other being non-polar, and they're just so different, and oil being hydrophobic. So those are some things that you should know. Riv says, do we need to know the chemical composition of specific acids or bases, such as hydrochloric acid, ammonia? No, you won't have to memorize any of those. I mean, it's nice that you know them. They were on the web quest, like hydrochloric acid, gastric fluid is HCl, table salt is NaCl, water is H2O. You should be familiar with some of the compounds that we've covered in the chapter. But I don't think I'm going to be asking you any specific questions like, here's this chemical formula, what compound is this? Won't be anything like that.
Can you see me?
Any other questions from section 2? All right, again, if you have any more questions on section 2, just ask them at the end of the live stream when I do the open Q&A. All right, we're going to move on to section 3. This one is on organic compounds. So specifically when we talk about organic compounds, we are talking about compounds that contain at least the element carbon. Quite often they contain carbon as well as hydrogen and oxygen, and even sometimes nitrogen and phosphorus. So carbon's kind of like the overarching theme. When it comes to organic compounds, that means they have carbon. So make sure you're good with that word. We do use organic when speaking about produce and fruits and vegetables and other crops, but that's not how we use it in chemistry. All right, make sure you can identify the difference between a monomer and a polymer. So monomers are the subunits that will build and kind of attach to each other and become polymers. So kind of use that prefix, that math prefix, mono meaning one, poly meaning many, so monomers and polymers. And the process by which monomers are grouped and assembled into polymers is called polymerization. So to give you guys an example, glucose is a monosaccharide, glucose is a monomer, but when several glucose units are attached to each other, they form polysaccharides, which are polymers. So think of monomers and polymers like Legos. An individual Lego block is a monomer. Multiple Lego blocks attached to each other build things, right? Larger units, those would be the polymers. Sometimes I've even used the example of a chain. An individual link to a chain is a monomer. The entire chain length, several different chain links together, that would be the polymer. That's kind of how that is. All right, so let's move specifically to the first of the 4 macromolecules. Let's start with carbohydrates. So these macromolecules are composed of carbon, hydrogen, and oxygen in a very specific ratio of 1 to 2 to 1. So C6H12O6 can be reduced to a ratio of 1 to 2 to 1, carbon, hydrogen, oxygen. That is glucose, right? C6H12O6. So what's important to know about carbohydrates? Number 1, they are instant energy. Our cells can break them down very, very effectively, very fast. It doesn't take much for them to do that. So that's their number 1 function. The next thing is they can also be used as structural components in cells. Specifically, we talked about cellulose, which is a polysaccharide found in plant cell walls. Gives it structure, gives it integrity, gives it protection. It's a good barrier for plant cells. So we talked about that. And also, in some cases, short-term energy storage, like we talked about with glycogen and how humans, our livers produce glycogen from glucose, temporarily storing it in our livers, temporarily storing it in our muscle cells. Now, if those glycogen units don't get used, then obviously the glycogen will be turned into something totally different in the storage, in the long-term storage of energy. We'll talk about lipids in a second. So some examples of monosaccharides, which are monomer units of sugars. We talked about glucose, that's number 1. Another example would be fructose. That's another monosaccharide. We talked about other types of saccharides as well. On slide number 23, there's an explanation of polysaccharides. These are the larger macromolecules of several collections of glucose units. So glycogen, for example, an animal-created disaccharide. Lactose, another animal-created polysaccharide, right? Mammals produce lactose. Well, not all mammals, but dairy animals like cows and sheep and stuff like that. So we talked about glycogen, we talked about lactose. Other polysaccharides like amylose, which is a starch found in potatoes and things like that. I think that does it actually for carbohydrates. Anyways, if you want to see the chemical structure, it's on page 23. It highlights all of that. And of course, starches are what plants do when they need to store energy long term. Plants don't store fat, they store starch. So they take all of those individual glucose units and they attach them, polymerize them to form starches, then they store them for usage over like longer winters and stuff like that. Because they can't photosynthesize during the winter. Moving on to slide 24, talks about lipids. So just like carbohydrates, we have carbon, hydrogen, oxygen. They are also important in energy, but they're more for long-term energy, so long-term storage of energy units, especially in animals. They're also really important cell membrane components. We talked very specifically about phospholipids, the phospholipid bilayer of the cell membrane. Now, you guys probably remember, these terms as it pertains to the lab report. So these are terms that we've covered already. Lipids are hydrophobic. Lipids are nonpolar, so they're not soluble in water. Some examples of lipids would be oils, whether it's like vegetable oil, corn oil, olive oil. Fats, which could be saturated or unsaturated. Cholesterols, steroids, waxes, like the waxy cuticle of plants on leaves. The monomer units of the lipid are the fatty acid and the glycerol. And to distinguish unsaturated and saturated fat, saturated fats are solid at room temperature. Unsaturated fats are liquid at room temperature. And unsaturated fats come from plant material, and saturated fats derive from animals. So that would be the main difference between those. The next macromolecule is the nucleic acid. So these are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Their main purpose is to store and transmit genetic information. They code for our characteristics and traits. Think about DNA and RNA as perfect examples of nucleic acids. Our DNA is stored in the nucleus of our cells, so that's where you would find nucleic acids. Nucleic acids are important in coding for our proteins, which also express our traits. So make sure that you look at the chemical structure of nucleic acids. You look at RNA, it's single-stranded. It uses ribose sugar. You have DNA, which is double-stranded. It uses deoxyribose sugar. That's where their names are derived from. The monomer unit of the nucleic acid is the nucleotide. The nucleotide has 3 specific subunits. So you have the phosphate group, you have the nitrogen base, and you have the 5-carbon pentose sugar. All right, so for proteins, we're going to transition away from nucleic acids. Proteins contain the elements carbon, hydrogen, oxygen, and nitrogen. The monomer unit is the amino acid. Sometimes we call proteins polypeptides because it takes peptide bonds to connect the monomer aminos to become peptides or polypeptides. Proteins are really, really important in forming and membrane components. So not only does the cell membrane have lipids, it also has proteins. They're really important biological catalysts. Remember the term enzymes, that's going to be a big part of the test. They're important in immune system support. They're also important in cell transport, vesicular transport, cell signaling, and communication from one cell to another. I gave you guys some examples of some different proteins: collagen, keratin, melatonin, melanin, serotonin. A couple are escaping my mind right now. Those are all different. Elastin, that was another one. Insulin, hemoglobin. These are all different proteins that do different things that your body produces. Remember, they take these amino acids and they build them into huge, enormous protein structures that your body needs. Now, there are 20 different amino acids, so obviously there's going to be endless amounts of protein structures. Huge varieties in proteins and what they can do and what they're for. Just keep in mind that amino acids are the monomer units, and the cell structures that assemble the aminos into proteins, so I'm talking about the polymerization of aminos into proteins, those are called ribosomes. Ribosomes are free-floating in the cytoplasm of the cell, assembling proteins all day long. That's what they do. That's their duty. So always associate proteins with ribosomes. That is their job. That is their functionality. So the DNA, the RNA in a cell, is coding, providing the instructions for assembling the aminos into the proteins that the cell needs. So the elastin, the keratin, the serotonin, the melatonin, all those things that your body needs to produce, it's all being assembled by your ribosomes using the amino acids. And the instructions are in the DNA and the RNA. So that covers all of section 3 on organic compounds and macromolecules. I'm going to look in the live stream and see if you guys have any questions. Regan asked, do we need to know the diagram for lipids, carbs, nucleic acids, and proteins? Now, the diagram I did on the board, that was just a study tool to help you guys organize structural and functional differences between each of the 4 macromolecules. So that's just a study tool. That's just a table. It's just a diagram. You're not going to have a diagram like that on the test. The test is going to have multiple choice questions that you have to, answer based on the best choice of your selections that are in that question. So no, there won't be any diagrams, but there will be questions about carbohydrates. There will be questions about lipids and stuff like that. Riv asked, do we need to know examples of monosaccharides, disaccharides, and polysaccharides? Yeah, you will. For monosaccharides, the most important one to know is glucose. Also make sure you know its chemical formula. So C6H12O6. For disaccharides, just know that is 2 sugar groups attached to each other. So technically, by definition, it does make it a polymer. So take, for example, sucrose. Sucrose is a disaccharide. That is a glucose and a fructose group chemically bonded together, a disaccharide. Glycogen is an animal produced disaccharide. Amylose, starch, those are polysaccharides. Cellulose is a polysaccharide. Lactose is a polysaccharide. Liv asks, would that chart thingy we made in class on Friday be a good tool to use for studying the section? It's a great tool because that has all of the chemical structures, all the elemental compositions, all of the functional for each of the functions for each of the macromolecules. It has examples. It has the monomer units. It has everything on there. So it's pretty good. It's a good way to go. Liv asks, do we have extended responses on this test? I don't remember actually. If there is extended response, there might be one, or actually I think it might be 2 short responses, like 1 or 2 sentences to answer. So I can't say for sure if there is extended response. I know there's a couple of written responses though, but I think they're short answer. Jack says, aren't carbohydrates C6H12O6? Well, that is an example of a carbohydrate. Specifically, that is a monosaccharide known as glucose, the most important of all monosaccharides. So is it a carbohydrate? Yes. Is it a monosaccharide? Yes. Is it glucose? Yes. Any other questions on section 3? And again, if you do have questions, just ask them at the end in the open Q&A. All right, so let's go ahead and transition over to section 4. Let's see here. This one is on chemical reactions and enzymes. So chemical reactions result in the transformation of a set of chemicals into a new set. So they undergo a chemical interaction, the bonds break, and then they reform. And anytime there's a chemical reaction, there are always indications that a reaction has taken place. So some indicators that a reaction have taken place, there's going to be heat produced, so it could be flame, could just be a change in temperature. There might be an odor, smell like something has happened. Color changes, greens and reds and oranges and browns. You'll see that colors are kind of morphing. You also see sometimes light is produced as a result of chemical reactions. So, we're talking like, take for the example of striking a match. When you strike a match on that side of the book or that book of matches, you have all of those things. You get a heat change, you get a smell, you can smell like the sulfur, you can see a color change, you can see the light is produced. All of those things show that there were chemical reactions that took place. Make sure that you know that in a chemical reaction, on the left-hand side of the reaction, you have the reactants, and then to the right side, you have the products. So there's a good example of that on slide 28. You'll see that it is the photosynthetic equation. It has the reactants on the left, CCO2, carbon dioxide, plus 6H2O, water, with sunlight, and that yields the products. C6H12O6, which is glucose, and then 6O2. Notice that everything that has happened there, you still have the same elements. They've just transformed and gone through some chemical changes. That is a great example of a chemical reaction, or actually several reactions that take place. Activation energy, we talked about how activation energy is the energy required for a chemical reaction to kind of proceed. So if chemical reactions are going to happen, if they're going to be efficient, activation energy has to be considered. So we talked about things like endo and ectothermic reactions, or exergonic, endergonic, that's synonymous. Just make sure that when you see those graphs, if it's an exergonic reaction, you're saying and you're implying that energy is being released. Therefore, energy is lost. So you would have energy starting at this level, and then at the end of the reaction, the energy level has dropped. That means the energy has been released out of the system. If it's endergonic or endothermic, the reaction starts with X amount of energy, and then at the end, energy is absorbed. Therefore, it kind of shows you that it starts at point A and then it's higher at point B. So that would show that energy is being absorbed or taken in. So on slide 29, you'll see those different graphs explaining that topic. On slide 30, specifically, this is on enzymes. So enzymes are biological catalysts. There are so many different types of catalysts as it pertains to chemistry. Enzymes are specifically biological catalysts. So within our cells, biological catalysts, enzymes, are what kind of help chemical reactions to take place in a more efficient manner. So the reason for this is enzymes serve a very specific function. Reduce the amount of activation energy needed so that chemical reactions can be completed efficiently and quickly and completely. So it's important to get the most products out of all your chemical reactions. So if you look on page 30, you'll see all the different So on slide number 30, you'll see all of the different chemical reactions with or without an enzyme. You'll see that without the enzyme, you'll see that the activation energy is very high. That's going to make the reaction very difficult to complete, and it's also going to make it difficult to proceed. So if you add the enzyme, that activation energy level can drop, which makes the chemical reaction more efficient, and actually you'll get more products out of it because it'll proceed a lot better. So think of biological catalysts, think of enzymes as kind of like jump starters. They allow the reaction to get going faster, and they allow the reactions to proceed without any hindrance. They make them more efficient, more products come out of it, and it just makes for a more efficient process. Specifically, we also talked about enzymes and substrates as it pertains to the lock and key model. That's on slide 31. So in the lock and key model, you have this kind of, it's just basically, I don't know, I'd say it's just an analogy, right? So you have the substrate, which has a certain shape, and then you have the enzyme, which has a certain shape. If the conformation is there and they fit, we can actually get conformation, that binding site, the active site between the substrate and the enzyme. If they fit, if they are compatible, then the products can be released from the enzyme binding with the substrate. So I'll give you guys an example of this. Let's say you have lactose, and let's say you have lactase. These two compounds are supposed to be for each other. So they fit. There is a conformation. They actually have an active site that confirms, hey, this active site works with this enzyme. Boom, it works. And then the products are released after that conformation occurs. So we use the example of lactose and lactase. If somebody cannot produce the lactase enzyme, we know that they are lactose intolerant. They don't produce that enzyme, they can't digest lactose. That's why so many people that are lactose intolerant have to find an alternative to drinking dairy milk. They have to drink coconut milk or almond milk or Lactaid, which is lactose-free milk, because their body doesn't produce the lactase enzyme. If your body produces lactase enzyme, you can digest lactose milk. So there's an example of how this binding between lactase and lactose can get the digestion process to proceed effectively. Now, some people are just sensitive to milk. They produce a little bit of lactase, but just not enough to where they can drink copious amounts of milk and other dairy products. So that kind of is the description of the lock and key model. You guys can see that on page 31, slide 31. And the last thing from that slide, you will see that there are three major factors that affect enzyme activity. Number one is temperature. There is a specific range of temperatures that certain enzymes function very well at. Take, for example, an enzyme in the human body. The human body's core temperature is about 99 degrees Fahrenheit. So I want you guys to think about what that represents in Celsius. I think that's like 37 or 38 degrees Celsius, I'm not sure. Usually it's about that range. So if an enzyme works well in the human body, we know that it has a limited range or optimal range in which it's going to work effectively. So if you go out of that range, the effectiveness of the enzyme becomes compromised. So if it's too cold or even too hot, the functionality of the enzyme becomes altered. Maybe it becomes less efficient, maybe it becomes less effective, maybe it becomes completely obsolete and it doesn't function at all. So if an enzyme is designed to work at a certain temperature, once it leaves that range, that optimal range, it can be useless pretty much. So you guys can see that curve kind of forms that bell-shaped curve. So optimal range, less optimal when it's too hot or too cold. Another factor that affects enzyme activity is pH. Obviously, they have a certain pH that is ideal, the optimal range of pH for certain enzymes. Take, for example, a digestive enzyme in your stomach. The pH of your stomach, your gastric fluid, is very, very acidic. Therefore, enzymes that help in digestion have to work well at very low pHs. So a digestive enzyme has an optimal range that works well between like pH of 1 to 3. Once it starts leaving that range of 1 to 3 and it starts going 4, 5, 6, the effectiveness of that enzyme starts to decrease. Therefore, those enzymes are almost rendered useless at basic pH levels. So a stomach enzyme, if it were placed in a solution that has a pH of 9 or 10, it's not going to be as effective. So, anything that functions well like an enzyme in your blood. Enzymes in our blood, they're in a solution of pH of about 6 or 7. So they don't work well whenever they're subjected to pHs of 1 or 2. So there is that optimal range for pH. And the last one is substrate concentration. You guys can see on slide 31 that there's a certain saturation point. Like, yeah, more substrate is good. We get more chemical reactions to take place, more products. But there comes a saturation point. Once it reaches that saturation point, it doesn't help to have more, it just kind of levels off. So you can see all those different diagrams there. And a perfect example of what you might see on the test, on slide 32, you will see a very similar question to what you see there. It's going to ask you, what is the optimal range of enzyme X or enzyme Y as it pertains to either temperature, pH, or substrate concentration. I don't know what form you're going to get, but each form uses a different factor. One of the forms I know is temperature, another form is pH, and another form is substrate concentration. So that's what you could look forward to there. So that's pretty much all of section 4. So I'll go to the live chat and I'll see if anybody has any questions there. Jack says, is there a good way to remember which graph is which? Not really, because, well, okay, so on those graphs, if you look at the temperature graph, it's a bell-shaped curve. If you look at the pH graph, it's a bell-shaped curve. So temperature and pH work the same way. If the pH or the temperature is too low, or if the pH or temperature is too high, the enzyme may not function properly, or may not function at all. So those two are similar. If you look at the substrate concentration one, that one is the only one that shows the saturation point, and then it just kind of levels off. So that's the one that's different. Temperature and pH behave the same. The substrate concentration is the one that kind of like tapers off a little bit, stops, kind of slows down the increase, and then it just levels. So that's a good way to remember those. Jack is asking endo and ectothermic reactions. So endothermic reactions absorb energy. The energy that it starts with is lower than the energy that it ends with. Because energy is being absorbed into the system, the end point has a higher amount of energy. You could see starting is here, ending is here. That's an endothermic reaction. You start with this much energy, you end with this much, right? So that's absorbing, taking in, adding energy. The exothermic reaction graph, you start with this much, you end with this much. So that means during the reactions, energy was lost. So the energy level is lower than it was when it started. Riv says, can we review the reason why an exothermic reaction is most likely to occur? You don't really have to know that for the test. It's not a specific question that I'll be asking. But if you're just curious and you want to know, combustion reactions usually, are used to describe exothermic reactions as an example. And because they, combustion is this spontaneous reaction between reactants, and it's more likely to occur. So think about like a gasoline engine. Think about, like cars, how they have combustion engines. Boats, diesel engines, they all work the same way. Those are good examples of exothermic reactions. The energy is being released from the system. But you don't have to know that for the test. You might just have to be able to know the difference between an ecto or an exergonic reaction, and then an endo or an endergonic reaction. So any other questions on section 4? Chemical reactions, enzymes, biological catalysts, things like that, factors that affect enzymes. And if nobody has any questions on section 4, then we could just leave the last few minutes here for just, I guess, an open Q&A. Anything from section 1, 2, 3, or 4. Or any questions from the lab, or any questions from your assignments, whatever you guys want to ask. Okay, so Jack asked, why are hydrogen ions positive? So I gave the example of water. When hydrogen ions dissociate in water, remember, water has 2 H atoms and 1 oxygen atom. When it comes to the chemical bonds that form between hydrogens and oxygens, the valence electrons spend more time around the oxygen. Because they spend more time around the oxygen, that side is slightly negative. More time spent on the oxygen. More electrons, more time, more negative. That means if they're spending more time around the oxygen, they're spending less time at the hydrogens. So that means those hydrogens are going to be slightly positive. And when acids dissociate in water, they form H plus ions. And when bases dissociate in water, they form OH minus hydroxide ions. So that's why we associate hydrogen ions as H plus. They form positive ions when they dissociate in water. Riv says, can we quickly jump through monomer units? No problem. So the way I like to think of monomers are like the chain, like the individual links of a chain, and all of the chains that, or all of the links of the chain that are attached to form the chain, that would be a polymer. So a monomer unit is a subunit. The polymer is a multiple unit compound. So a monosaccharide is a monomer. A polysaccharide is a polymer. So glucose is a monomer. 5 glucose units attached to each other is amylose, a starch, a polysaccharide. Cellulose is a polysaccharide. Lactose is a polysaccharide. Many, many subunits attached to each other. And the process by which monomers are assembled into polymers is polymerization. Another example of that would be like amino acid, right? 1 amino acid, 20 amino acids making a protein, a polymer. Abby asks, will we have to label the enzyme catalyzed diagram? You might. Depends on your form. Look at the there's I forget what slide it is. On slide 31, there are probably 5 or 6 questions on the test on that slide. At least 5 questions on slide 31 on the test. So it might be, a few multiple choice questions. You might get one of those diagrams, one of those graphs. You might see the diagram where you see the enzyme and the substrate complex, where it shows the products and it shows the enzyme and it shows the binding site or the active site. You may have to label it. I don't know. It depends on your form. So I would say that slide 31 is very critical to know for the exam. That's a good question. Riv, I don't think I understand your question, honestly. For monomers, I meant jump through the monomers of polymers. Could you reword your question? I'm not really sure what you want me to clarify. Okay, so Abby asked a nice question. This one's pretty good. I'm not sure if this is on the test, honestly. It might be on a form or two, and it might not be. I'm not sure. So, the order in which your cells utilize energy for macromolecules. The first thing that your cells look for energy are carbohydrates. So we're talking about like in the sequence of available energy molecules. The first thing your cells want, they want to get the energy from the carbs. Once your carbohydrates are completely utilized and they're completely used, the next thing they go to are lipids. So you've used all your carbohydrates, they switch to start searching for energy in the form of lipids. So this is where your fat burning comes from. Burn your carbs, the next thing is going to be fat. So let's say you've used all your carbs, you've used all your fats. The last thing that could be utilized for energy is proteins. That is your cell's last resort. They don't want to use proteins for energy. They want to use proteins for structural components, membrane components, immune system support, rebuilding muscle tissue, repair, maintenance, and the damage of muscles and things like that. They don't want to use proteins primarily for energy. They want to use carbohydrates, then lipids, and proteins as a last resort. But I will say this. Of those 3 macromolecules, notice I left out nucleic acids because they have no energy value. That is strictly for the coding of proteins and genetic inheritance. Carbohydrates provide 4 calories per gram. Proteins provide 4 calories per gram. So the most efficient when it comes to calories and energy are lipids. They can actually produce the most potential energy. 9 calories per gram of lipids. So a lot of animals that are hunters, a lot of predatory, apex predator animals, they choose to hunt animals that are very high in fat content because they contain a lot of lipids and they have a lot of potential energy. So apex predators love fatty animals. And Riv, if you want to ask your question tomorrow before the test, just to get clarification, that would be fine. But we won't have much time. We're going to be taking the exam pretty much within a couple of minutes. I'll take attendance. You can ask your question before we start the test, but it's got to be quick. We do have picture day tomorrow, y'all. So I think we're 10:15 to 10:30 is our window. So make sure you're aware of that. Pictures. Any other questions from chapter 2? Looks like we are an hour into the live stream, so I really only wanted to spend 30 minutes, but hey, if you guys needed the help, that's fine with me. Jack, I'm not eating dinner because you asked so many questions. I don't get dinner today, so thank you for that. It's all your fault, Jack. You owe me a dinner. I like ribeye steaks. I know, Hayden. It's all his fault, too. I get no dinner. I'm going to just drink some water and go to sleep. And think about the dinner I could have had. Jack, you ain't eating steak right now, man. You ain't eating no steak. Liar. Alright, any other questions? Hayden, you wanted to watch me eat wings? Oh my goodness. Any other questions on chapter 2? I know, Regan. Thank you for feeling for me. I appreciate that. We're good? Any other remaining questions? Regan, stop it with the pudding. Any other remaining questions you guys can put in the comments section below. I think I'm set to be notified. I may be able to answer some questions before I go to bed, but probably not. I'm pretty tired. Have those questions in the morning, first thing before we start the test. That's it. I hope you guys were able to benefit from this review. Hopefully you guys do pretty well on this test. You guys worked pretty hard this week and last week, and you should see the benefits. So that's it. I'll see you guys tomorrow.